Okay, it's 8:01, and for the purpose of maintaining all of us on time, I'm going to proceed, and I'm going to be sharing with you some slides because I think that this is it's totally new and important um, information um, to share with our patients. So I'm just going to go to slideshow, and I'm going to go from the beginning. And is everyone seeing this slideshow? Okay, so this is work that has been done since the year 2003. And this is me in the last 40 years. I have learned very well to do the interventions that I'm going to share with you to maintain my youthful look. And right now I'm professor of dermatology at UConn and director of the Mohs Fellowship at UConn. I'm still Senior Vice President of the Skin Cancer Foundation and still have my private practice in New Canaan, Connecticut. I'm a board certified dermatologist with boards also in immunodermatology and over 100 publications, chapters, and textbooks. And so as the world changes and all the colors of the beige, white, beige, and browns get intermixed in the world, and the world becomes a more brown and more beige, we need to kind of learn if the aging process is similar in different ethnicities. And how to do this, it was a very interesting project that was started by Procter & Gamble. And let me just put it here. It was started by Procter & Gamble in 2003. So all this project was funded by Procter & Gamble, was done by the scientists at Procter & Gamble in collaboration with Alexa Kimball in Harvard. And the objective was to see and understand the fundamental molecular mechanisms which contribute to the skin aging, both intrinsic and extrinsic, using different ages, body sites, and ethnicity. And as you can see in the slide, multiple decades of patients or participants were included from the age of 20 to the age of 70. Multiple sites of biopsy sites were selected. Face and arms being the exposed areas and buttocks being the covered areas. And multiple ethnicities, there were patients that were Caucasian, African Americans, Chinese, and Hispanics. And multiple measures were used: the appearance of the skin structure, the biology, the DNA, genomics, proteomics, that I will be sharing with you. Here you see a kind of a distribution of the participants. The largest number of participants that you can see are the Caucasians and the African Americans. That was the original study. And then subsequently, the Hispanics and the Chinese were added. And you can see that there is in all decades been 20 to 24, 40 to 44, 60 to 64. And the genetic analysis, you're going to ask me, how the heck these people know that there was African American and Caucasian? So it was 23andMe was utilized, in which the genetic components were identified, and for the patient to be for the participant to be included as Caucasian has to be in the more than 74% of Northern European or European origin. For a participant to be considered totally African American, has to be from West Africa or Africa in a very high percentage of 85% or more. And you can see here the histology. You remember that I told you that the biopsies were done in the face, the arms, and on the buttocks. So exposed areas, being that in here the face, you can see that in the Caucasian patients, you can start seeing in the face, which is the most exposed area, you can start seeing elastosis, which progresses along the different decades. Where you can see in the African American, there is no evidence of elastosis in any of the decades evaluated, including the 60s. You're going to start seeing in the Hispanics so elastosis, significant elastosis starting in the 60s, as well as in the Chinese population. So therefore, you can conclude in here that elastosis is seen earlier in the Caucasian population in the 40s, never seen or documented in the African American population, and seen in the Hispanic and Chinese population in the exposed area more in the 60s. So it will be 20 years after the Caucasians. And what is the importance of documenting histologically elastosis? And this slide can show you that obviously, if you have elastosis, you look old. If you don't have elastosis, your skin looks supple. If you have elastosis, your skin sags and there is folds and wrinkles. If you don't have elastosis, your skin is supple. So a marker of the aging process histologically is elastosis. And when we compare the African Americans to the Caucasians in the perception, the facial appearance of the skin, you can see that for both 64 years of age, you can see the African American looks 10 years younger than the Caucasian. So there is a discrepancy of at least in the appearance of a decade between the African Americans and the Caucasians. And what about gene expression? Remember, what we're analyzing here is we take, for every gene, there's messenger RNA that then produce a protein. What we're taking is really the messenger RNA and seeing the proteins that are produced or not produced. And then we're going to expose the genomics. And here in this slide, which is a busy slide, but the only thing that I want to remind you is anything in a heat map that is the genomic expression, anything red is increased expression. Anything blue is decreased expression. So in here, you can see all Caucasians, African Americans, Hispanics, and Chinese. And you can see the different decades. And this is the epidermis, and this is the dermis. And in general, as it says in here, what you can find is that in the epidermis, there is 75% of the genes will have an increased expression with aging, meaning by that it's turning into red. So in the epidermis, as we age, most of all the groups are going to show an increased expression of genes. While in contrast, in the dermis, you see that is blue. Blue is decreased expression. While in contrast, in the dermis, you can see that the majority of the genes, 75% of dermal genes, decreased expression with the aging process. So let's go individually to each decade and see and kind of teaser or delucidate or dissect what are the different pathways that are affected at different decades. By that, we can kind of intend to intervene and try to affect the aging process at different decades differently. So and I'm going to share with you the different genes that are involved. The first gene that it's noted to be start to be defective is the natural antioxidants or the property of natural antioxidant, and is seen to be start to be affected in the 20s. In the 30s, cell energy is noted to be affected. In the 40s, cellular senescence. In the 50s, skin barrier. And in the 60s, all the matrix products. Now we're going to at least take one gene from each one of these decades and these expressions and see what happens. So for the gene expression for natural antioxidancy, it was selected the gamma glutamyl cysteine synthase gene, which is a natural antioxidant in the skin. When you start looking at the age of 20, you start noticing that it starts at and across the decades, there is a decrease in the expression of this gene as we mature, as we age. So the dysfunction of a natural antioxidancy of the skin starts at the age of 20. What about, I told you that in the decade of the 30s, the genes that are starting to get dysfunctional are the genes of cell energy. We all know that the mitochondria is the structure in our cells that is kind of the battery, maintains all the energy for our cells. And so in the mitochondrial structure, we can find membranes, matrix, lumen, ribosomes, and is in the function of it is mainly electron transfer chain, oxidative phosphorylation. So all the energy transformation and storage resides in the mitochondria. And this is the compartment that seems to be starting to be defective in the 30s. In here, you can see that there is some components that you can add to reverse that dysfunction. You can use niacinamide that we know very well, and I will share with you other properties of niacinamide. Upregulates filaggrin, and it also impedes the transfer of melanosomes from the melanocytes to the keratinocytes, improving pigmentation. And there's also peptides. We can see the PKTTKS peptides that can also increase the energy pathway in the mitochondria that start being dysfunctional in the decades of the 30s. What about cellular senescence? I told you that in the decade of the 40s, cellular senescence starts to be dysfunctional. The gene that we're following for cellular senescence is the CDKN2A, which really codes for P16. And this is a marker for senescence, cellular senescence. If we evaluate the epidermis separated from the dermis, and we see the expression of cellular senescence across the different decades, and we evaluate the exposed areas, in red the arm, and in blue the face, and compared in contrast to the buttocks, that it's a covered, it's non-exposed. You can see that cellular senescence is in the epidermis is really affected mainly in the sun-exposed areas, meaning by that there is a direct impact of the sun exposure in inducing the dysfunction into the cellular senescence. Also, you can see it happening in the dermis. So it's not exclusively of the epidermis. Also in the dermis, you can see that there is an effect or an impact of sun exposure increasing cellular senescence across decades, mainly in the sun-exposed areas in contrast with the sun-protected areas. You can see some change in the sun-protected areas in the dermis, but not to the significance that on the areas of sun exposure. So what about epidermal moisture barrier, the gene that was selected to be followed for the function of moisture barrier, or the function of the barrier, is the lamellar body, and the gene is Eugene gene. You know that the lamellar body is a product in the granular layer that gets extruded into the intercellular space before the cells lose the keratinocytes lose their nuclei, and in this area there is ceramides and sphingolipids, and this is what is going to join the epidermal cells and create a functional barrier. So if we follow the lamellar body through the coding, through the following the function of the Eugene gene, and we followed it in the exposed areas in the arm, which is in pink, and we followed in the buttocks, which is sun protection, we can see that there is a decrease in both the exposed and non-exposed area. So there is an intrinsic and extrinsic component of dysfunction that starts at the 50th decade in the epidermal barrier. And what about autophagy? Autophagy is a function of a cell, is kind of the recycling of the cell when the cell is non-functional anymore. There, all the organelles get kind of digested and broken down in pieces, and then it's kind of being thrown to be recycled for the next cell, for the next progenitor cells to use that material. So autophagy, let's see how sun exposure affects autophagy. It's seen here in the autophagy genes that the autophagy genes are mainly affected in sun-exposed areas and not as much in the exposed areas, at least when we take the UG gene expression. However, when we look at autophagy and compare, see all the different lifestyles that include sun protection, moisture of the skin, aerobic exercise, and smoking. And we compared the people that don't use sun protection in pink, the people that use sun protection in blue, and across the decades, we can see that the people that use sun protection in blue demonstrate a better maintenance of the autophagy genes than the people that do not use sun protection. So therefore, the autophagy genes are affected negatively by sun exposure. If the person uses moisture in the skin frequently, frequent use of sun moisture is in green, and not frequently is in pink, you can see that a person that uses moisture demonstrate that it maintains the function of the autophagy genes more than a person that does not use moisture. If the person does regular aerobic exercise frequently, it's in green and not in pink, you can see that when you do exercise regularly, you can maintain the autophagy genes function better than if you don't. And moreover, if you smoke, here is in pink versus not smoking in green, you can see that the autophagy genes are negatively impacted by smoke. So all the lifestyle factors are going to negatively affect the autophagy genes. Again, explaining that autophagy is kind of the recycling of all this, of all the components of the cell that is dying. And ultimately, after menopause, you can see that there is an increment in the aging process at almost 7 times than before menopause. And we know from the work of, from previous work, that estrogen has an impact into the function of the epidermis and the dermis, work done by Tanya Phillips. And it is actually very much affected after menopause. So in this brief presentation, I was able to show you that there are similar trends in all ethnicities, but there are subtle differences that in skin changes across the different 4 ethnicities, guaranteeing that we need more investigation. We demonstrated that transcriptional analysis shows significant changes in the epidermal and dermal gene expression across multiple pathways starting early in the 20th decade. And UV exposure strongly affects skin biology and influences the changes of gene expression, including genes related to senescence, autophagy, transcriptional regulation, kind of indicating that if for no other reason we would like to have some protection in our skin, if it's not for the purpose of protecting us from skin cancer, then it will be for the protection of against the aging process. And now I would like to, before I continue, I would like to open the floor for a discussion of what is your takeout message from this presentation, and what is your experience? Well, since I don't hear anyone sharing their experience, I'm going to proceed with the next part of the presentation, which is mainly the part about skin cancer that it's also important to know. And in here, I'm going to just present you the component of the Hispanics that is the one that I have studied the most. I have just followed the Hispanic population for the last a decade or so in the United States, and knowing that the Hispanics used to be 35 million in the year 2000. And right now, in the year 2020, we're approximately 60 million. So there was an increment of 48% from 2000 to 2011. And now there's been another increment of approximately another 10 million in the last decades. And then remembering that these Hispanics are not only foreign born, that is 36% of them, but there are 64% are born in the United States. And is a population that is a young population. If you notice here, 57% of the Hispanic married couples have children under the age of 18. So it's a young population that is projected to be to form that is right now a compromise 18% of the whole population of the United States. And where are these Hispanics? Mainly they are of Mexican origin, 64% of them, followed by the Puerto Ricans and the Cubans and the Central Americans. And they are located mainly in California, Texas, Florida, New York, and Illinois. But it's now evident that as you look at these dark colors, that there is a distribution of Hispanics in the year 2020, mainly in the Southwest and in the South. But the major 5 cities that contain the majority of the population of Hispanics is California and Texas. Nearly half of 47% of Hispanics live in California and Texas. And these are the skin cancers that the Hispanics actually develop. These are squamous cell carcinomas, basal cell carcinomas, and melanomas. And these are melanomas of superficial spreading acral lentiginous, and then also of nodular. And melanoma is the 3rd most common cancer in the United States, and is the most common cancer in these ages between 25 and 29, and second in the ages of 15 to 29. The incidence of melanoma in Hispanics is between the one that is seen in Caucasians, which is 21.6 per 100,000, and 4.5 per 100,000 for the Hispanics, being the lowest in the Asian and the African American population. Unfortunately, even though the incidence is lower, the early diagnosis is also lower. So the early diagnosis, which is the curative state, is seen less in the African American population. Only 48% is diagnosed early, 74% in Hispanics, while most of the majority of the Caucasians are diagnosed in early stage. Most of the Hispanics and Caucasians develop superficial spreading melanomas. And in the Japanese and in the Black population is acral lentiginous melanoma. But what was to my surprise was the fact that in the past 2 decades, the incidence of melanoma in Hispanics has risen by 20%. And it tends to be a population that is younger at diagnosis and has more advanced tumors, have thicker tumors that lead to regional involvement and distant metastases, having a worse survival as compared to whites. And this has been evident in this presentation, there is one study done in Florida, and you're going to see that the two focus of the high incidence of melanoma in Hispanics are basically Florida and California. And this was a study that was done in melanoma cases of known stage and ethnicity between the years of 1990 to 2004, obtained from the Florida Cancer Data System, evaluating 41,000 cases of melanoma, in which it was demonstrated that the incidence of melanoma increased in white Hispanics to an 18%, and also in the African American to a 26%. The incidence increased in the white Hispanics, but what is more dramatic is the distant metastasis and the regional and distant metastasis in the Hispanic and the African American populations. And another important fact, if you do evaluate the melanomas among non-whites in Florida and compared to other places in the United States, it was demonstrated in this paper that the incidence of melanoma among Hispanic males was higher, 20%, than compared to the other counterparts in other locations. So could ultraviolet light be mediating this increased incidence? And there's some evidence that might be the exposure, but what I'm trying to bring in here is what will be the reason that there is that increased incidence? Is there more exposure? Is there lack of understanding that exposure to the sun leads to skin cancer? And in this study that was done in a clinic that took care of different ethnicity, and it included Asians, African American, and Hispanics. It was asked, questions of behavior under the sun, knowledge about skin cancer, if they had recreational sun exposure, if they use sun protection, if they teach sun protection to their children. And although 43% of the subjects really knew that they had sunburn, only 35% of them perceived that there is any association of sunburn and the developing of skin cancer. And even though they knew that they had burning, they really did not know that they had potential for skin cancer. And moreover, a third of the ethnic skin patients or the parents never used sunscreen, and then perpetuated this behavior in their children. As I told you, most of the epidemic of melanomas in Hispanics is happening in California and in Florida. And you can see in this study that in high school students of white Hispanics compared to white non-Hispanics, if you take the two populations which have comparable phenotypes and you ask them, do you get yourself exposed to the sun? Do you use sunblock? Do you perform self-exams? You're going to find that in the white Hispanics, there is less perception of wearing or practice of wearing sun protective clothing. 60% of less performed self-skin exam. Less than 70% are taught how to perform skin exam, and they don't think about the chances that they have to develop skin cancer. And they have more tanning. They tend more than twice, 2.5 times that the rest of the white non-Hispanics. If you do the same questions in the area in California, for example, you're going to find the same behavior. It's white Hispanics are more likely to be tan, less likely to do self-examination, twice or more of using tanning beds in the past year. And they have no perception, even though they burn, they have no perception that they can get skin cancer. And self-examination is likely to be less than 60% in that population. So obviously, is the perception in the population that they are not prone to skin cancer has to be change, because this population definitively is at risk of developing skin cancer. If you take the whole population of the United States and you evaluate for the incidence of melanoma, you can see that the Hispanics have a significantly lower incidence than the Caucasians. However, the Hispanics are diagnosed in a younger medial age, at a later stage, and thicker tumors, and so leading to a worse prognosis. And this is mainly seen in patients in California and Florida. And it seems that is also in these two locations, seems to be associated with lower socioeconomical status. And these melanomas are mainly of a nodular type, as in contrast with the superficial spreading that is the most common in other Hispanics in other locations. So now I have taken you, very briefly through why there is a disproportionate high mortality of melanomas in Hispanic as compared to Caucasians. And we know that there's delayed in diagnosis and treatment. The delayed in diagnosis and treatment might be directly related maybe to the misconception of the people of the same, the Hispanics, and the misconception of the doctors that this population is not at risk to develop skin cancer. Maybe there is less access that is culturally and linguistically appropriate for these patients. And there is also less access to medical care. And so I think that we can redeem this or remedy this if we teach the physicians that really Hispanics can develop skin cancer, teach the young Hispanics at the level of middle school and high school that they really could be prone to skin cancer, and practice safety precautionary measures early in life. And maybe we can also make a medical care, not that we're in the advent of telemedicine. Maybe telemedicine could help us to get to those distant populations that have no access whatsoever to medical evaluation. We can help these patients or these Hispanics to get some access via telemedicine, implementation of telemedicine. So I think that not only I've shown you before that sun exposure can be associated with dysfunction of the skin from the point of aging, but also has shown increased incidence of basal cell carcinomas and melanomas in some Hispanic patients in California and Florida. And maybe we want to decrease that incidence. Maybe we should all promote sun safety practices with protective clothing, protective sun protection hats, sunglasses, and if for nothing else, for the aging process or for the development of skin cancer. And you can contact me either in my private practice or at UConn in these phones and these emails. And thank you for your attention. Now I'm just going to stop sharing my slides and hear any comments that you can share with me. Okay, I have a question here. Do you have favorite sunscreens that you recommend to patients of color? Actually, and I have no interest whatsoever, monetarily or otherwise, but I know that Pearl Grimes has done some work demonstrating, that the visible light has an impact into skin types 4 and 5, inducing hyperpigmentation. That's visible light. And that this visible light is only blocked by ferrous oxide. And ferrous oxide is in some tinted sunblocks, but we don't know the percentage of it. And the percentage that she recommends to be the most effective is in the 3% range. So there's a product that is made by Dermablend, and is a product of, I think that they have like 40 different shades, and it's made with increments of percentages of iron oxide. And so if you go to your favorite pharmacy, or store, beauty store, you can ask for Dermablend and all the different shades, and they will find the shade that is appropriate for you, and they will find the shade that has the highest concentration of iron oxide, specifically intended for patients of skin of color, to prevent the visible light hyperpigmentation, induced hyperpigmentation. Okay, Renee Scott, how can you tell the difference between hematoma and melanoma in darker skin types? Well, actually, that's a very good point. But that's one of the things that, as we in dermatology evolve to the utilization of dermatoscopes, we become very much experts in seeing the signs of melanoma and the pigment distribution, versus a hematoma that you're going to see collection of blood that it's more purple. So with a dermatoscope, you can do it. Sometimes in a simple visual evaluation might be a little different, but we as dermatologists are very keen in the evaluation, at least those of us who have a lot of training in skin of color evaluation of melanomas in skin of color. Okay. Okay. Thank you. Somebody, Gita Ahua says, very informative. Thank you very much. Solal from South Africa says hello to everyone. Oh, and please make sure that if you have any questions, I gave you my 2 emails, my personal and the practice email. I did not give you the Yukon email because I received so many emails there that I can only deal with Yukon at Yukon. But so if you want to contact me, you can contact me to my personal email or my office email, or you can call me, and the phone numbers are available in my last slide. So feel free to contact me if you have any questions and I could be of any help. And I want to reiterate that, these seminars and networkings are available to us by funding from Pfizer, that we really very much thank for their funding of these very educational meetings. And also they are made possible by the Society of Skin of Color, that I encourage you to join us, because now more than ever, we need as more exposure and collaboration as possible from all the sources of skin of color. And also, if Veronica can also, please, you can also contact me through the website of skinofcolorsociety.org. And for those of you, I have one here that Yvonne Nong, that told me that if I can be able to give the earlier part of the presentation somewhere. If you contact me either by email or phone, I will be able to share them with you. Okay. So Veronica?
Yes, I'm here.
Okay. Let me answer this one. Nicotinamide anti-aging cream, could you give us more details? Okay. Niacinamide as an anti-aging cream, as you know, I told you that it can improve the energy pathway in the mitochondria. It also increases the filaggrin production, decreases the transfer of melanosomes from melanocytes to keratinocytes. So niacinamide is contained in many products. You just have to look at it as an ingredient. I know that some of the Olay products and no interest in monetary interest in it, but all the Olay products contain niacinamide, as well as there is niacinamide in 5%. And there's many of the compounding pharmacies that put niacinamide into their products. So if you want the kind of the combination that I consider to be the best combination for anti-aging, you need niacinamide, you need peptides of different sources, pentapeptides, decapeptides, you need hyaluronic acid, you need some form of retinol, and some form of ceramide. So if you have all these components in some plus or minus form with one or different anti-aging creams, you're working in favor of your skin. And for those that are postmenopausal, and I don't have any interest in this product either, but there is a product that is called Emipel. Emipel contains an artificial plant origin and a synthetic peptide that gets associated with the estrogen receptors in the epidermis, but then gets degraded immediately and does not go into the general circulation. And so that product is used as an adjuvant for the postmenopausal woman. Moreover, the product also contains niacinamide peptides and all the other components. So it's for the postmenopausal woman is a very good product to use. Okay, going back to the first part of your presentation, how might your research finding be translated in the clinic to facilitate patient education? Okay, so this is the way to make it kind of practical for the bedside or for the clinic side. So what I tell my patients is that the sun interferes with the normal function of the skin and make us age because there's different compartments of our skin that are going to not produce the appropriate proteins that then make our skin better. So to interfere with that, I tell them that all the ingredients that I just told you that are good for the reversal of that induced dysfunction. And not only I tell them, you know, like, for example, the antioxidant, not only they should apply antioxidant like vitamin C to their skin, they should also ingest it. Like niacinamide, not only they should apply the niacinamide in the skin, they should also ingest it because niacinamide is being demonstrated to also protect you against sun damage, systemic sun damage, used as oral sun protection. So I try to also intervene a little bit about, you know, not only things that they apply and the sunblocks that they use, but also the lifestyle decisions and selections, demonstrating that if, you know, there are genes that are affected by our lifestyle selections, then maybe we should just select the right lifestyles to improve our health in general. Well, any more questions before we depart in this beautiful evening? Well, Veronica, I think that we have accomplished what we wanted to accomplish tonight, and let's sum up if you can help me with the departure.
Sure, I think we did too. Thank you. Excellent job, Dr. Perez. Everybody, thank you so much for joining us tonight for the Pfizer Regional Networking Symposium. I hope you enjoyed it, and we hope to see you at future events over the next couple of months, virtually and in person eventually. If you have any general comments or feedback about the webinar, then feel free to email info@skinofcolorsociety.org. And we look forward to seeing you at a future event. And we thank Pfizer for supporting and sponsoring this event and SOTS. Have a good night, everyone. Thank you very much.
Good night.